And so now I'd like to introduce Matthew Simon from the Jackson Laboratory, and can't wait to see his data.
All righty. Hopefully the mic will behave for me. Thank you, Catherine, for the introduction. And I also want to give a big thank you to the SRF, Michael, and everybody here for the opportunity to come and discuss the new mouse model that we're making on behalf of the SYNGAP community. And I just want to take a quick minute to really emphasize the importance of this community. It is no small thing to try to tackle something like SYNGAP1 by yourself. It is a group effort. And I hold the philosophy that one of the key things about us as human beings is we have literally moved mountains because we work together, because we come together and every little individual effort adds up to just monumental changes. So for everybody here, I hope you understand that you are a part of that effort and should take great pride in that, because I know I do. But with that out of the way, I want to talk about the new Q503 star mouse that we are currently phenotyping on behalf of the SRF. But a quick little bit about me and my group. So I'm part of the Jackson Laboratories Rare Disease Translational Center. So we view ourselves as a patient-centric disease modeling and preclinical testing organization. So we're relatively new to the Jackson Laboratories, but we saw that there was a very large unmet need for the rare disease communities and kind of bringing together a lot of the loose ends and the very difficult things that I know many people here as part of these foundations know that's kind of hard to bring together. So when we talk to individuals, families, and foundations who are dealing with a rare disease and need to build more resources to move the research as well as the therapeutic testing forward, we kind of use this pipeline that I'm showing you here, where we start by identifying the patient mutations themselves. And right after we have identified that, the very next step for us is to anticipate a genetic therapy. This is the big difference, I think, that we kind of bring is we don't build the mouse model until we've already got an idea of how we would use that mouse model for therapeutic screening and testing. So this goes into the initial design of the animal, which is then the next step. And right now we're in this nice little teal green step for this new mouse, where we are phenotyping the animals, making sure that they have, patient reflective behaviors as well as biomarkers. And we can also use these animals once that's validated for initial therapeutic testing and screening to identify chief candidates and then support that moving into preclinical data that supports IND submissions to the FDA. So this is not a comprehensive list, but this is what we have currently available publicly for a lot of the mouse models that have SYNGAP1 mutations or can be used for SYNGAP1 research. Now, of course, we want to make sure that a lot of these models do already have phenotypes that are associated with SYNGAP1. Probably the big workhorse or the work mouse here is our SYNGAP1 knockout. But more recently, there's been several new patient-derived alleles that have been characterized and donated. And this table is already out of date because we've already heard about some amazing and really excellent new work, especially with the sleep as well as the EEG phenotypes. And ultimately, I hope that this table a year from now has a lot more little green markers on it and is much more built out. But I'm going to talk a little bit more about one of the newer models that we've generated, which is Q503. And this is a patient-derived allele. It is an early stop nonsense mutation. So basically causes the RNA transcript to go through nonsense-mediated decay, and we get the classic haploinsufficiency. Now, like many of the mouse models that we've already developed for SYNGAP1 and that others have worked with, we usually start out by using the Black6 background. But as many of you are probably aware, Black6 pure background doesn't breed particularly well. We usually then have to cross these animals into a 129 background, which I think is an important factor to kind of remember when we are working with mice as models for humans. Mice are inbred. They're very almost like a clone of each other. So they don't have as much of the little fine tuning that we see within the human population. And I'm kind of excited to mention that there's a lot of new tools that are currently being developed, not only at the Jackson Laboratories, but at other institutions that are leveraging a lot of the new AI technology, as well as some, really robust bioinformatics to kind of do a better job of screening initially what might be the better background to put a disease model into and not just necessarily start with the Black 6. So in our case, we did start with Black 6 and then moved it into the 129. And then just by crossing this to a B6129F1, we're able to generate our cohort animals, which include our litter mate controls. And for the current data I'm going to talk about today, which is the first chunk of the phenotyping that we've done, we started out by looking at these animals a little bit later into their life. So this is around 20 to 24 weeks of age, and this is where most of the data I'm going to show you is reflected. But we did go through a large number of validations as well as characterization of these animals. And I will point out that a lot of this is based on all the wonderful work that's already been done in other models. We basically got to go through and say, that assay seemed to work there, that assay seemed to work there. We'll see how that works in this particular mouse model. So one of the first things we needed to do was to validate the molecular aspects of the animal. So we looked at RNA expression and protein abundance. We also do basic monitoring of the animals, not only for the health of the animals, since at the Jackson Laboratories, we are very, very much one of the leaders in showing how well to treat animals used for animal research. But we also monitor body weight and survival. And then lastly, we threw a number of behavioral assays based on what we already know from other mouse models about SYNGAP1 at these animals to see how they stacked up to what's currently available and to get some decent characterization on it. So right away for the molecular characterization, and what I'm showing you here, and for most of these presentations, I am showing you data that is got error bars that are standard deviation, not SEM, so don't be intimidated by their size. The RNA expression in whole brain extracts was reduced by about 50% in both males and females. When we looked to spinal cord and other tissues, really quickly, the abundance of SYNGAP drops off, so resolution's kind of lost there. However, when we look at protein abundance, both in brain and spinal cord, we found that in both sexes, our SYNGAP1 mice do have roughly that 50% reduction, so supporting the haploinsufficiency of the model. So with that out of the way, we then began looking more at the animal itself. And as I mentioned, body weight is one of the things that we always like to look at with brand new mouse models. We never quite know how things might be affected there. We didn't see any particular issues with early lethality or any other attritions with the heterozygotes. Homozygotes are non-viable, much like other mouse models for SYNGAP1. And while the males didn't show much of a body weight change, we did see there was a difference in the females. And currently, we're following this up with our secondary phenotyping cohort. The females were roughly about 20 to 25% heavier than their wild-type litter mates. Now, there could be a couple of reasons for why this might be, and this is also part of our follow-up work that we're doing, which is it could be elevated appetite, metabolic differences, or changes in the body mass distribution. So we're planning on looking at that with our next batch of mice that we are running through, but it was just kind of an interesting little result that we saw, which was this sex-specific thing. And as you'll see later, there are some other assays where we do have an effect in one sex, but not the other. Now, we also wanted to look at the YMA spontaneous alternation. There's been some previous work in other models. They did show there was a difference there. However, in our model, we did not see any particular change for the spontaneous alternation rate. But we did see that there was an elevated number of entries into arms. While this doesn't indicate anything about the working memory of the animals, what it does indicate is that the animals are being a bit hyperactive. Now, this is also reflected what we saw with open field metrics, which also showed here, as you can see, a very beautiful result in that both males and females are much, much more hyperactive than their litter mate controls. And I will tell you, as an individual who does these mouse models with the idea of doing therapeutic testing involved, this is the kind of data that we really love to see off these models. This very large differential means that we can test therapeutics and have really good power for seeing how they're moving that needle back towards the healthy end of the spectrum without having to run an obscene number of animals for these type of studies. We also saw that the animals had elevated vertical activity, which kind of go along with that increased hyperactivity and distance moved. And where we started to see another little interesting thing here was the amount of time the animals spent in the center of the arena. Now, mice as prey animals don't like to be in the center of anything. All the predators can spot you out there, so they tend to hug the walls. So it's kind of non-stereotypical mouse behavior when they tend to hang out more towards the center of an open space versus hugging the walls there. And while there was a trend with the males, it didn't quite meet statistical significance, but we did see it was very robust with the females there, indicating that they might have a little bit of change in how their normal behavior is elicited. Now, we also looked at novel object recognition. So this is kind of more of a working memory assay. And here we found that both the males and the females did have a curious preference for the novel object more than their wild type litter mates. And this is also reflected in the discrimination index. So in general, they found the novel object much, much more fascinating than their siblings who weren't carrying a mutation. We also did look at the three-chamber social approach. And here we saw some sex-specific differences as well. Now, right off the bat, we didn't see a major differences in either sexes as far as their preference for the stranger versus the object. Both the wild type and the Syngap mice did have a preference to hanging out with the stranger more than just the object alone. However, when we looked a little bit closer, which is how much time they were spending with the novel or with the mouse, the stranger, we found that there was a difference when it came to the males. Here, the males had a lower discrimination index. So while they did tend to be around the other mouse more often than the object, their actual interaction time with that mouse was reduced. So it seems that they prefer the company, but not necessarily direct interaction. Now, another aspect we wanted to look into was the sensomotor gating. And for this, we used acoustic startle and pre-pulse inhibition. So right off the bat here, we saw another change in the distribution between the sexes, whereas the females had a pretty profound startle response. Basically, they flinch much more and they have a higher amplitude of this response compared to the wild type animals. This wasn't seen in the males. And likewise, when we looked at the pre-pulse inhibition, we found that the females had a deficit here. Essentially, you put an initial pulse out that's a little bit more quieter than the next pulse that startles them. And usually you would see a reduction in that. It's kind of if you hear somebody walking down the hall and all of a sudden they open your door, you're not as startled as if somebody just burst in on you. But for the SYNGAP1 mice here, the females at least, they didn't seem to have that gating. So essentially, even with that little bit of pre-pulse, they were still startling pretty heavily when they give them the more louder startle pulse. And then kind of lastly, from this first round of phenotyping that we've done, we also wanted to look at the sleep disturbances in these animals to see if they did recapitulate what we've seen in some other models. And here is where we also saw that there were some major differences, but specifically right around the night sleep cycle. So mice are a little bit more active at night, so this is kind of their wake time. And here we found that both the males and the females spent a lot less time sleeping than the litter mates. And their bout lengths were reduced as well. So these animals basically were not catching the power naps quite as well as their littermate controls did. And during the daytime, when they're typically a little bit less active, again, they seem to be catching up on some of their sleep during that time, but there was no major differences with the bouts, which would indicate that they're not really having, sleep disturbances during their normal sleep time, but not picking up as much sleep during the daytime when they normally would be a little bit more active. And this might speak a bit more to their hyperactivity being a major role and why that sleep cycle might be changed there. So that kind of wraps up what we've done initially. We have a whole nother cohort or two actually coming along to test some other metrics. But so far, hopefully I've been able to show you that we've got a whole bunch of these things already kind of identified in these animals and characterized. And just to kind of sum up here, we have confirmed that we do have our haploinsufficiency via the protein reduction in both sexes. We've seen hyperactivity is a very beautiful readout in these animals for both sexes. and that there are some specific differences for both the acoustic startle as well as their 3-chamber discrimination index. And on top of all that, we do see that there are some sleep differences as well as some novelty preference differences in these animals, which goes along pretty well with what we've seen in other knockout models of this particular gene. Now, moving forward, as I mentioned, we have additional cohorts that we are testing. Our first one was done at kind of later ages. We're now redoing some of these assays at younger ages, around 8 to 12 weeks of age, just to see if there is any major differences. And also, again, from a therapeutic testing point of view, if we see that some of these phenotypes are still there and still just as robust earlier on, that's beautiful. It means you can test and you don't have to wait for the animals to age out before you can see whether or not you've actually moved that needle towards healthy. We're also looking into some biomarker discovery to see if we can identify, some more molecular mechanisms or molecular readouts that we can use to inform upon the therapeutic testing studies. And lastly, one of the big things that we're doing with the next big batch of animals that we have is we're going to be looking into the EEG and EMG seizure and sleep phenotypes to get a better idea of how these animals are impacted on both of those metrics. And so lastly, I do kind of want to talk really briefly about, this overall process for those of you who are new to therapeutic testing or using these type of model systems. The path to the clinic does require several things. And fortunately, we already have a lot of these for SYNGAP1 and specifically with this particular model. So first thing you have to have for a mouse model is you have to validate it. Does it have patient-related phenotypes? Here we do. We have several of them that we can work with. And secondly, as I mentioned, do you have large differentials with these models and their wild type controls? This is also very important so that you can run these therapeutic screening studies without having to use a boatload of mice. And here we have that as well. What we're currently working on and would be nice, but not necessary to have is to have disease relevant biomarkers. I will tell you the FDA and other regulatory agencies absolutely adore these. If you can find a biomarker that is related to that disease and you can find it not only in the human, but you can find it in the mouse, that is a great readout. Those also tend to be very, very tight as far as their data goes. So you can run even fewer animals and screen a lot more therapeutics a lot faster. And when you present that data to the FDA or to the regulatory agencies as you petition for a clinical trial, they can be very, very robust in helping make sure that they say yes, because they know that you have a readout that doesn't require anything particularly invasive for the patient and will tell you very quickly whether or not your treatment is working in the human being. And lastly, of course, the things you would need for the clinic is you do need a candidate therapeutic to start with. And using animal models or cell models, you can usually get pretty far with your initial work, which requires you to screen for safety and efficacy, make sure that the therapeutic doesn't have a negative reaction and that it's actually doing something. And then from there, you can refine things such as the route of administration and dose, so that when you're ready to put together a pre-IND letter to the FDA, you have all this information, you have a platform, you have a therapeutic, you have safety and efficacy, you have everything that really says, we've done as much as we can without going into a human being up until this point. We need to start talking about moving into the human being. And so with that, I'm going to give a quick shout out to the entire team at the Jackson Laboratories, but again, also to everybody here for being able to attend. As I mentioned earlier, I think bringing together all this talent, all these dedicated individuals, these families, and I really want to emphasize what they were saying earlier in the clinical talks. The families, I think you guys are so important. I mean, I can sit here and look at a mouse all day. I can look at reports and read papers and scientific journals that will tell me what we should expect to see from these mechanisms. But what is actually happening in the patient? And for rare diseases, when we sometimes have patients who number in maybe the teens at best, parents and clinicians telling us what they are seeing is some of the most informative data when no other data is available. So never underestimate just how important that is, and just being able to say what that experience with your children, with what you're dealing with, what your challenges are, that is so important for us to be able to work together as a group to overcome these diseases. And with that, I thank you all for your time. Move on to questions.